Welcome to Biotanemy. Programmed cell death. Programmed cell death, or PCD, is a regulated biological process in which cells undergo self-destruction in a controlled manner as part of normal development, tissue homeostasis, or response to stress. Types of PCDs are 1. Apoptosis, or classical programmed cell death. 2. Necroptosis, or regulated necrosis. 3. Pyroptosis, or inflammatory cell death. 4. Ferroptosis, or iron-dependent cell death. 5. Autophagy-dependent cell death. 6. Parthenotos, or PRP-dependent cell death. 7. Entosis, or cell cannibalism. So, let us see about apoptosis. Apoptosis is divided into different types based on the origin of the triggering signals and the molecular pathways involved. They are 1. Intrinsic apoptosis, or mitochondrial pathway. The pathway begins with cellular stress or damage. Let's talk from the point of p53 activation. Normally, p53 is found in the cells bound with MDM2 protein, which guides it to its gradual degradation. During DNA damage, p53 gets released from the MDM2. The free p53 will inhibit the anti-apoptotic proteins like BCL2, which leads to the activation of pro-apoptotic proteins like BAX or BAC. Activated pro-apoptotic proteins will oligomerize on the mitochondrial membrane, permeabilizing the outer membrane of the mitochondria, leading to the release of cytochrome C to the cytoplasm. Also leads to the release of SMAC proteins, or second mitochondria-derived activator of caspases, also known as Diablo or direct IAP binding protein with low PI. The SMAC protein will block IAPs, or inhibitors of apoptosis proteins, that are capable of blocking caspases. Cytochrome C binds APAF1, or apoptotic protease activating factor 1. APAF1 will recruit procaspase 9, which leads to the formation of the apoptosome complex. Apoptosome activates initiator caspase 9, through cleaving the procaspase 9, which then activates the executioner caspases, caspase 3, and caspase 7. The executioner caspases will cleave nuclear and cytoplasmic substrates. 2. Extrinsic apoptosis, or death receptor pathway. As the name suggests, the death receptor pathway begins with the binding of death ligands like CD95L, TNF-alpha, TRAIL, etc. to their respective receptors, leading to their activation. Let's go with CD95L to CD95, also known as FAS. Activation of FAS will recruit the adaptor proteins, FADD, or FAS-associated death domain protein. Together they form a DISC, or Death-Inducing signaling complex. The DISC will bind with procaspase 8 and cleave it to activated caspase 8, or initiator caspase. Activated caspase 8 works in two ways. A. They can either directly activate apoptosis executioner caspases like caspase 3, 6, and 7. B. Or they will cleave pro-apoptotic protein BID to form truncated BID, which can be directly translocated into mitochondria, triggering cytochrome release. 3. Perforin, or Granzyme-Mediated apoptosis. It is also known as immune cell-mediated apoptosis. The process starts with cytotoxic T cells, or natural killer cells, recognizing infected or cancerous cells via MHC-1 presentation or stress ligands. They will form an immunological synapse with the target cells and release cytotoxic granules that contain perforin and granzymes into the immune synapse. Perforin polymerizes in the target cell's membrane to create a transmembrane pore, which allows the granzyme entry into the cytoplasm. Granzymes can directly cleave the caspase 3 and 7 to initiate apoptosis, or it can activate the intrinsic pathway by cleaving BID protein. Granzyme A can cause DNA damage via single-stranded breaks. 4. Stress-Induced apoptosis. The stress apoptosis occurs when misfolded proteins accumulate in the endoplasmic reticulum. When it occurs, the cell will trigger the unfolded protein response, or UPR, to restore the normal cell function, and will move on to apoptosis if it fails. UPR will operate mainly through three proteins. Let's only see apoptosis. A PERK, or protein kinase, are like kinase pathway. PERK is an membrane protein that senses unfolded proteins. On activation, it increases the translation of specific stress-related proteins, like ATF4 or activating transcription factor 4. ATF4 will induce CHOP protein, or C/EBP homologous protein, which is a pro-apoptotic transcription factor. It inhibits anti-apoptotic proteins like BCL2, promoting pro-apoptotic proteins like BAX and BAK. B. IRE1 or inositol, requiring enzyme 1 pathway. IRE1 is a transmembrane sensor in the ER. In normal conditions, it is kept inactivated in the ER bound to the chaperone protein BIP. During stress, the chaperone gets dissociated from IRE1. The free IRE1 activates the JNK protein or CJUN and terminal kinase, which phosphorylates and inhibits BCL2. See ATF6 pathway or activating transcription factor 6 pathway. ATF6 is an ER resident protein. Under stress, it is transported to the Golgi apparatus, where it gets cleaved by proteases to release its cytoplasmic active form. From the viewpoint of apoptosis, it will activate the CHOP protein and enter into the apoptosis process. 5. Anoikis or detachment-induced apoptosis. Normal epithelial cells rely on integrin-mediated adhesion to the ECM for survival. Detachment leads to the disruption of integrin signaling, particularly pathways like FAK or focal adhesion kinase and PI3K/AKT that promote survival are disrupted. Let's go with the PI3K/AKT pathway. When this pathway is disrupted, the AKT or protein kinase B will remain inactivated without phosphorylation. So, it can no longer phosphorylate FOXO or forkhead box O protein and deactivate it. The active FOXO will be translocated into the nucleus and promote the transcription of pro-apoptotic proteins like BIM, PUMA, and NOXA. Increased levels of BM protein will inhibit BCL2, leading to the activation of BAX/BAK proteins. Those are the different types of apoptosis. Let's end it in here. We can continue to discuss about other forms of programmed cell death in the next video to avoid confusion and weariness.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.